One other question that we have is, why do we have claim on genomic integrity maintenance? We have an insight as to the impact of the culture system on genomic integrity. That's a bold claim. So basically we checked the genome of the cells going in the system and the genome of the cells after amplification going out of system. And basically we have a match, so we maintain the genomic integrity. That's something that we should have. In vivo, the pluripotent stem cells, they ought to maintain the genomic integrity, because if there is a problem, then the whole body is going to inherit it. So there's a huge pressure to do a good job there. And basically in classical culture systems, you actually see mutations accruing over time. So that's a very important thing to check for. And I would say that the usual suspect as to why we have that drift is that there is a lot of cell death. And if you have a mutation that gives you a survival advantage, you're going to be selected for, especially if there is a lot of cell death. If you don't have much cell death, you're not going to have the opportunity to be selected for.